Hello everyone, this is Mrs. Chandana Santosh Nirali, Assistant Professor from Food Chemistry and Nutrition Department, KK College of Food Technology. Under course titled Biochemistry course number FCN 112, credits to 1 plus 1, we will see regarding protein structure. It includes amino acids, polypeptide levels of structure. Monomer. Monomer is nothing but single unit. The single unit that makes up a protein is amino acid. Based on its name, which two functional groups would be found in an amino acid? Obviously, amino group as well as carboxylic group. An amino acid is sometimes referred to as residue in general. Amino acid structure. 4 components attached to a central carbon which is known as chiral carbon. Likewise, amino group, carboxylic group, hydrogen atom, variable R chain, that is side chain. Determine which functional group is acidic and basic. Explain how you know. Acidic is COO minus, basic is NH3 plus. Depending on their nature, we know the nature. Amino acid structure. Amphiprotic, containing both acidic as well as basic functional groups. The ionized form is absorbed in aqueous solutions because the acidic group can donate H plus ions to the basic group. In normal, they will be unionized form, whereas when they are get dissociated in water, they will show the ionic form, that is COO minus NH3 plus, as the COOH group donates its H ion to NH2 group. Amino acid R group side chains. Differences in R groups produces 20 different amino acids. Among them, 8 are essential as body cannot synthesize them. So, there are 8 essential amino acids. Beans rich in lysine, grains rich in methionine. How to remember? Pasta e fagioli. Pasta and beans, a healthy Italian soup. Whatever the alphabet is denoting one amino acid of these 8 essential amino acids. This is a trick to remember easily the 8 essential amino acids. Amino acid R group side chains. Physical and chemical characteristics of the R group determine the unique characteristic of an amino acid. Amino acids are classified into 4 groups based on their R group: nonpolar, polar, acidic, basic. Activity. If we see, given the 20 amino acids, group them into 4 categories based on the properties of their R groups. Those 4 categories are nonpolar, polar, acidic, basic. Nonpolar amino acids has hydrophobic R group. For example, R group might be hydrogen CH3, CH3CH3, CH2 CH, CH3 CH3, CH, CH2, CH3, CH3, which are present glycine, alanine, valine, leucine, isoleucine respectively. Nonpolar, methionine, phenylalanine, tryptophan, proline. Polar R group, hydrophilic they will be. Example, serine, threonine, cysteine, tyrosine, asparagine, glutamine. Amino acids with the charged functional groups at cellular pH can be either acidic or basic. Under acidic, aspartic acid, glutamic acid. Under basic, lysine, arginine, histidine. These are electrically charged amino acids. Condensation reaction to join two amino acids requires carboxylic group, amine group. One amino acid is carboxylic group, the other amino acid is amine group, condenses and forms a peptide bond. What is the name of the new functional group formed? It is known as peptide bond. Peptide bond, link between amino acids. Ester bond it is. Protein shape determines function. Amino acid order determines the shape conformation of a protein. Conformation determines function. Function depends on its ability to recognize and bind a molecule. So, amino acids, conformation, function, binding are the components important. Antibodies bind to particular foreign substance that fit their binding sites. Enzymes recognize and bind to specific substrates, facilitating a chemical reaction, biochemical reaction. Neurotransmitters pass signals from one cell to another by binding to receptor site on proteins in the receiving cell. Levels of proteins, if you see, there are four types: primary, secondary, tertiary. These three organizes folding within a single polypeptide, whereas the fourth category, quaternary, at interaction between two or more polypeptides that make a protein. Primary structure, unique sequence of amino acids, sequence determined by DNA. A slight change in primary structure can affect a protein's conformation and ability to function even. Primary structure, it will be a linear chain. Example, sickle cell anemia. Abnormal hemoglobin developed because of a single amino acid substitution causes hemoglobin to crystallize, deforming red blood cells and leading to clots in blood. Only single amino acid position change causes sickle cell anemia, where the shape of the hemoglobin molecule will be lost and it will be sickle in shape, and it causes a diseased condition. Secondary structure results from hydrogen bonds at regular intervals along the polypeptide backbone. Typical shapes: alpha helix coils, beta pleated sheets, folds not found in all proteins. Tertiary structure, interaction between R group and R groups and backbone. Tertiary structure, types of interaction: hydrogen bond, ionic bond, hydrophobic interactions often in interior of protein, covalent bonds like a disulfide bridge formed between the sulfide groups of cysteine amino acids. Tertiary structure, proline kink. Proline is the only amino acid in which the R group is attached to the amino group, forms a natural kink in the polypeptide, helps to shape tertiary structure. Aggregation of two or more polypeptide subunits forms two types of proteins, globular as well as fibrous, not found in all proteins. It will be water soluble in nature, compact spherical, example hemoglobin. Quaternary structure, again fibrous category, will be water insoluble, thread like, example collagen. 3 polypeptide supercoiled like a rope provides a structural strength for role in connective tissue. Levels of protein structure: primary structure will be a linear chain, secondary structure will be combination of alpha helix as well as beta pleated sheets. These two together forms the tertiary structure, and the tertiary structure will be forming the quaternary structure. Protein folding occurs spontaneously, aided by chaperone proteins. Chaperonin provide ideal environment for folding. These are the different steps: hollow cylinder cap, chaperonin fully assembled, and there is an unfolded polypeptide enters the cylinder from one end. The cap attaches to the end, causing the cylinder to change shape in such a way that it creates a hydrophilic environment for the folding of the polypeptide. Later, the cap comes off and the properly folded protein is released outside for its functioning. Tutorial protein folding. The link you can follow so that you can see a visual add video. Conformational change. Changing the shape of a protein, reversible, does not disrupt a protein function but rather is what defines the protein's function. Change occurs in response to the physical and chemical conditions. Example of conformational change: carrier protein. Carrier protein will enter from one side. Obviously, it will be released to the other side because the carrier protein allows only the gate for the different molecules. Denaturation, a change in the shape of the protein that disrupts the protein function. Alteration in the environment, that is pH or salt concentration, temperature. It disrupts bonds and forces of attraction and causes denaturation. Normal protein to denatured protein, it can be reversible. Renaturation. Only some proteins can return to their functional shape after denaturation, but others cannot, especially in the crowded environment of the cell. Thank you very much for your patience listening.